Today we are going to start with one more topic, that is lac operon. What is lac operon? Lac stands for lactose sugar, and operon. What is operon? Operon is a group of genes which are structural genes along with some other genes which control the regulation of those structural genes is known as operon. For an example, in lac operon, the gene Z, lac Y and lac A. These three genes are the structural gene, whereas along with operator, promoter and the regulator gene, it forms a operon. So it is present on DNA segment. It is present on DNA. So now let's understand why it is required. Suppose there is a bacterium called E. coli. Now E. coli can grow in the medium containing normal sugar, that is glucose, or in absence of glucose, if lactose is available, the bacteria they have to switch on to the lactose digestion mode. So if lactose is absent, the enzymes responsible for the digestion of lactose are not required. But yes, if glucose is absent and lactose only is present in the medium, they need the enzyme which can digest the lactose sugar. So lactose sugar is digested by beta-galactosidase. So here the lactose sugar is an inducer. It induces the production of such enzyme which can digest itself. So let's understand the components of the lac operon first. First of all, the structural genes. Structural genes are genes which are to be transcribed and ultimately translated, and the enzymes or the proteins will be formed. One more. Adjacent to the structural gene, one more gene is here ZYA. Lac Z, lac Y and lac A are the structural gene. Adjacent to this structural gene is the operator gene. Now operator gene is the site where the repression molecule binds. We'll understand what is repression molecule. So operator can be turned on and turned off. When it will be turned on and when it will be turned off? It need to be turned on when the inducer, that means the lactose sugar, is present. So if the lactose is present, the genes, structural genes need to be transcribed. So in that condition, the operator gene is turned on. And if the lactose is absent, the operator gene will be turned off. Next to the operator is the promoter. So promoter gene is the site for the RNA polymerase enzyme to be bound. RNA polymerase, as we know, it is responsible for the transcription process. So once the RNA polymerase binds to this promoter site, it moves to the operator site. If it is turned on, it will again glide over or slide over and will transcribe this Z, Y and A gene and form the corresponding mRNA and ultimately the protein. Now, one more gene in the lac operon is regulator gene. Now, what is the function of regulator gene? Regulator gene is responsible for the production of specific protein that is known as repressor protein. So, it is responsible for the production of repressor protein. And what is the function of this repressor protein? Generally, in normal condition, suppose this blue colored protein is repressor protein, this protein will bind to this operator site. So suppose if it is attached to the operator site, now if the RNA polymerase enzyme will bind to this promoter site, suppose this is an enzyme, RNA polymerase, will it be glide over to this operator site? No, because it is already blocked, right? So this is turned off condition. So regulator gene responsible for the production of repressor protein which binds to the operator site and make it turn on, turn off. If it binds, it is turned off. So if the enzyme RNA polymerase binds to the promoter site, it will not be able to glide over to the operator site as the repressor protein is already bound to it. And ultimately the enzymes will not be formed. The transcription will not occur and ultimately the enzyme will not be formed. One more. If the lactose is present, what is the role of lactose sugar? Here, the lactose sugar acts as an inducer. Now, the inducer has affinity, the repressor protein, it has affinity towards the inducer. So, if the lactose is present in the medium, in that case, this repressor molecule, suppose this is a repressor molecule, it will bind to the inducer and it will form repressor-inducer complex. So, now, repressor is already busy with this inducer. So, now it is not available here to bind with the operator molecule. So, operator gene. So, now the operator gene is open. So, when the RNA polymerase will bind to the promoter site, operator site is already open. Why it is open? It is turned on because of the inducer, because of the lactose. Lactose is present and it is already fused with the repressor protein. So, repressor protein is not bound to operator site. Now, promoter site, already the enzyme is there, RNA polymerase. Now, RNA polymerase will glide over to operator and then it will turn on the structural gene, transcription of the structural gene. So, all three structural genes are transcribed. LAC-Z is responsible for the mRNA for beta-galactosidase enzyme, LAC-Y for permease and LAC-A for transacetylase. So, these three structural genes are responsible for the production of mRNA for different proteins. So, this way, when the lactose present and lactose absent, the operon is turned on and turned on. Now, let's go in little detail. Suppose this is a Petri plate and this Petri plate contains glucose. So, right now lactose is not present. So, now the E. coli bacteria is present in this. Now, in this E. coli bacteria, it has this operon in it, in its DNA, right? So, now if glucose is present, it does not require lactose operon to be turned on. So, in that condition, the regulator gene will continuously produce repressor protein, but here the lactose is absent. So, repressor protein is free to bind to the operator site. So, now it will bind to the operator site here. Once it binds to the operator site, this operator site is blocked. Now, what about the RNA polymerase? RNA polymerase cannot glide over to this operator site and next adjacent to it all the structural genes. So, the structural genes will not be transcribed and ultimately the operon is in off condition. Now, if the lactose sugar is present, if the sugar is present, the sugar which is there in the cytoplasm by the permease, it will increase the permeability of the membrane and it will reach to the cytoplasm and in the cytoplasm it will bind to the repressor molecule. Now, this repressor is already busy with the the inducer molecule. So, now this site, the operator site is totally free to bind for the RNA polymerase enzyme. So, RNA polymerase enzyme, it will glide over from here to here to here to here and will transcribe all the structural gene and LAC-Z will form beta-galactosidase, LAC-Y will form the mRNA for the permease and LAC-A will form the mRNA for transacetylase. So, beta-galactosidase, permease and transacetylase are the three mRNAs from which ultimately the respective proteins will be formed and the beta-galactosidase will digest the lactose sugar. Once all the lactose will be utilized, again the lactose which is bound to this repressor will also be utilized by the beta-galactosidase. So, now repressor is free, again it will bind to the operator site and the operon will be shut off. So, this is very simple and very easy theory of lac operon. Hope you liked it.